discuss about genetic basis of disease and some of their disorders. And I'm your coordinator, Dr. Kemwa. So I'm going to move the video, stop the video so that you can concentrate. So without much ado, diseases, as you are going to learn, have many etiologies. There are those that are 100% environmental, and there are those that are 100% genetic. And we are going to see the roles of genes in causation of human disease. On the right here, you can see this chart whereby by bad luck, if you are struck by lightning and you die, then the etiology for that problem is 100% environmental, as you may imagine. And down here, there are diseases that have 100% genetic causation, for example, Down syndrome, achondroplasia, and so forth, sickle cell anemia, and so forth. In between, there are diseases that are going to be a mixture of both environmental and genetic. For example, diabetes, whereby there is a role of genes and there's a role of the environment, also phenotype, weight, height, and all these. For example, if your genes are for a tall height, and then by bad luck, nutrition is bad, hence environment, then you're going to you are not going to get what your genes are getting. So that's why we are saying other phenotypes require both genetic and environmental factors. So class, today we are going to look at some of these disorders that have a purely genetic causation. Look at that long list. Definitely it's not conclusive, but you can see on the right here is a disease like Down syndrome. But look at all these: alpha thalassemia, blood disease, glycogen storage diseases, you learn this in biochemistry, beta thalassemia, hemophilia, you learn this in hematology, Canavan's disease, Marfan's, myotonic dystrophy, Tay-Sachs, and all these diseases that have a basis, that have a pure genetic basis. So let's look at how this arises. From your high school days, you do remember we have transcription and translation. So within in the nucleus, there are chromosomes, and these chromosomes contain DNA, and it's from this DNA that we generate protein. So the formation of DNA, the transcription of material from DNA to RNA, this is referred to as transcription, and it goes on in a process whereby there's a template of DNA where mRNA is formed, and from mRNA we find there is translation, that is the formation of proteins. And this you are going to see is that during all these, problems can occur in both the transcription and even the translation. And this is known as mutations. Okay. And feel free to stop me in between to ask questions so that you can get it clear. You can do it in the chat box, or optionally you can ask a question and I'll be pausing in between if there is any issues. And I'm going to assume that this is with you and you know this, or you're going to read more of this. And this is the thing we are talking about, that DNA is within the nucleus. And during the process of transcription, mRNA is formed. mRNA leaves the nucleus into the cytoplasm. And from there, within the ribosomes, you find that there is protein synthesis. So this is clear, and this is the process we are going to go through. And let me just remind you of a few things. That as human beings, we are made up of tissues, and these tissues are made up of cells, and it is within the nucleus of cells that we find these chromosomes. And these chromosomes are always in pairs. And as you all know that in our cells, we have 23 pairs, 22 which are autosomes, and 1 pair which are XY, which are chromosomes, sex chromosomes. And portions of these chromosomes is where we find genetic material. And from long time, from the 50s, through studies that are done by Watson and Crick, you know that this is in a double helix. And when you karyotype, you know, in karyotype is in the way in which we study chromosomes. We know that they are arranged from chromosome 1 to chromosome 22. Like in this particular case, you can see here, in a situation of Down syndrome, we have 3 chromosomes on position 21 as opposed to pairs. And in this karyotype, we know this is what is referred to as trisomy 21. There are 3 chromosomes on position 21, and this condition is referred to as Down syndrome. You can read more about this, and they are arranged from the largest to the smallest, which are the XY chromosomes, and this is referred to as karyotype. So to go back again, our cells have a cytoplasm, cytoplasmic membrane, and a nucleus. And it's within the nucleus that we find chromosomes. And these chromosomes, if you're going to pull it out, you know that they have a short end P and a long end Q. And if we pull them out, you'll find this DNA, which is usually put as in the form of a double helix. So a gene is a unit of DNA that codes for a specific protein. The location of a gene on the chromosome is referred to as a locus. And genes, not all part is specifically coded for. There are specific areas referred to as exons that contain the DNA code for the protein, and the intervening sequences of a known function which are referred to as introns. So you have to remove these introns. You have to cut them out and put the exons together in the mRNA before it leaves the nucleus. So during all this process is when we can have things that are referred to as mutations. Again, DNA can be thought of as a blueprint for an organism. It's composed of small molecules referred to as nucleotides. And remember that these nucleotides are distinguished by the 4 bases: adenine, A for adenine, C for cytosine, G for guanine, and T for thymine. So this DNA is a large molecule, or referred to as a polymer. And in nowadays, you can look at DNA as a form of digital information. And a single strand of DNA can be thought of as a string of 4 letters, ACGT. And remember that 3 of them code for an amino acid. And hence, if you look, all these CTG, GSC, these are coding for specific parts of a protein. And you can look at them as each containing an amino acid. Even when you look at this, any substitution of any of these 1 letter by any problem, any mutation, then you can see that whatever is going to be formed is going to be abnormal. And essentially, that is the basis of a disease. Again, to go back at it, if you stretch, if you look down here, this is the chromosome. This is a nucleus with various chromosomes. And you look at this chromosome, you stretch out all this, you'll find a double helix. And this double helix has various base pairs. And it's a certain length of this that will code for a certain protein. And when you look at them, there are those that are coded, those are coding for maybe some proteins. And in between, there are certain parts referred to as introns, so that when you are forming mRNA, you have to remove these introns and put together these exons for you to go now manufacture the protein. Is that clear? Before I go to some of these genetic, the events of some of these things, how this was discovered? Any questions? I'm going to pause. Kelsey, you can read some of the questions. I can see some of people are putting some things on the chat box. Can you read some of them that I can answer?
So far, there's been no questions in regards to the lecture topic. Oh, okay. Someone has just asked if introns have no function at all.
So introns are like if you are reading a book, let's say DNA is like a book, okay, and a book is organized in chapters and all this. So you can think of introns like some advertisements or something within the book, maybe making the book beautiful. But currently, introns, maybe other than structural, maybe like a structural part, they are thought not to have any part, because when you look at the protein, you'll find out that before mRNA is formed, those introns have to be removed. Then you put the exons together. You see, this is an exon, this is an exon, this is an exon. You have to put this together in the mRNA so that now when it goes to the ribosome, then you can form a protein. So introns currently, there are a lot of studies that have been done. But as you are going to see later on, during removal of these introns, sometimes the enzyme that is responsible for removal, for splicing out these introns, sometimes it may not be accurate. And hence, when you find some mutation, there's some things called splice site mutations. So that between that exon and that intron, if this enzyme that's responsible for removal of that intron, sometimes it may go a bit into the exon. So you get what is referred to as splice site mutations. So this is extra from what I've told you. Introns usually have to be removed, and the exons have to be put together during the formation of a protein. So currently, introns are thought to have no really function.
Okay, another student has asked, between introns and exons, which one contributes to the larger portion of the DNA?
So actually, the genes, if you look back, let me just go back. Maybe it's not there, but apparently the intervening introns, which make up most of the size of a gene, have unknown function. The exons are the fewer ones, and they're the ones which have function. The introns are big, but they usually don't have any function, and they make up most of the size of a gene. And the introns must be cut out, and the exons put together in mRNA before leaving the nucleus.
Okay, thanks. Another student has asked if you could pardon the last slide that you were on before you asked to answer questions.
That's the one you want?
Okay, I'm not sure. I think the student needs to just specify which slide. Yes, she said this slide.
So there are approximately 3.2 billion base pairs in every cell that build up the human genome, but the genes form only 1.5% of the human genome. Okay, so whatever has been studied, remember there's a lot of studies, there are geneticists, people are specialized in this gene and their functions. We have only discovered roughly 1.5% of the whole genes that form part of the genome. So there are a lot of material in there that have not been known, and these form part of the introns. And again, a gene is a segment of DNA that encodes for a protein. Can I proceed?
Oh, okay. Another question is to confirm that the wrong sequence of nucleotides causes mutations.
Yes, so if you look at this strand of DNA, a mutation is a change of any of these base pairs. So for example, Do you know amino acids? I guess you do. So there is one amino acid, let's say, one amino acid, let's say thyrine, and it's encoded by CTG. If you remove that G, this now becomes CTC. And now CTC is a different amino acid. So if you look, that's now called a mutation, removal of any of these base pairs, and then that removal results in a totally different protein, which most of the time could be either structurally, either in a 3D or 2D, it may be functionally incorrect, and we're going to see some of these proteins. So that either removal of a single or a segment of that, we are going to see how this occurs, is referred to as mutation. Okay, let's just proceed. You may ask others as we go along. In molecular biology, since the 50s, when Watson and Crick did studies about DNA, they found out that DNA is actually a double helical structure. After that, there have been many people who have studied and found out, for example, that ribosomes are organelles within which proteins are formed, and this was found by this guy called George-Emile Palade. That this Howard Tenmin and David Paltmore isolated enzymes, the so-called restriction enzymes. These are the enzymes that cleave, okay, that cleave parts of this DNA. And now DNA can be cut into reproducible pieces with these so-called restriction enzymes, also referred to as endonucleases. And this is the basis for recombinant DNA technology or gene cloning. So that now, especially in the plant, those who study plants, there's been a lot of recombinant technology in plants, whereby you can make, for example, bananas that can continuously keep reproducing. But similar studies in human beings have been restricted because of ethical issues. But enzymes, especially the restriction enzymes that can remove a piece of DNA, are now in use. And this has been studied from the 1970s. There are many of these, for example, introns and exons have been studied by these two gentlemen. And these are some of the things that have been going on until recently. In the 90s, we have had the Human Genome Project, whereby genes have now been mapped out, and the total, the human genome is already known. And you can read this in books like this, the book referred to as The Human Genome. It's written, whereby you can know what, let's say for example, what is, which genes are responsible for color, for height, where are they? And you can even go to a genetic laboratory and have your own genome sequenced out. So genetics right now is seen like a branch of IT, and we have doctors and scientists who are studying this. And now it's been found that you can do many multiple things. For example, I've told you about in plants. You can have tomatoes which are resistant to cold, and this is part of what is now being discussed about genetically modified foods or organisms, or those GMOs basically are based on that. That you can genetically modify, let's say, maize, and find a variety that can resist drought or can survive drought longer than the usual maize. But how this in our setup is being done, especially in the disease, is situations whereby genetic modification is being introduced, especially in certain metabolic diseases. This is not widespread, but it's being pushed for. For example, there are young people who have diabetes mellitus, and it's because they lack that gene that produces insulin. Now, it's actually possible to introduce to them a gene that is able to produce insulin. But the applicability of this is still on hold because of ethical issues, because once introduced, you never know what people are able to do with our genome. So let's go back again. What's a genotype? The genes that you have that represent a certain condition are referred to as the genotype. When it expresses itself, this is referred to as the phenotype. So that's what's clinically apparent. But remember, whatever you have as a gene, as a genotype, is not always presenting itself. This is because of the playing between the genotype and the environment. Pleiotropy refers to the appearance of multiple effects from one gene. So sometimes you find one gene that have multiple effects. This is referred to as pleiotropy. And what we have just been discussing is referred to as mutations. Mutations refers to alterations from the normal. So that if one gene has some mutation, then the resultant protein may be abnormal, okay? For example, factor 8, part of that factor 8, which is responsible for the clotting, when there's a mutation in it, then the factor 8 that is being produced doesn't cause, the clotting system is affected. Hence, you have hemophilias, people or patients who bleed for long because clotting has been affected because of a mutation in factor 8. So mutations refers to alterations from the normal. So examples of mutations that occur are, for example, sickle cell disease, which is a point mutation on chromosome 11p. Hemophilia, whereby we have a point mutation on X chromosome where factor 8 is being encoded for. Down syndrome, as I started, I told you, is a chromosomal abnormality at position at, chromosome 21, where we have 3 instead of 2. Duchenne's muscle dystrophy is a deletion at chromosome XP. Cystic fibrosis is a point mutation at position 7q. I've not put in the loci for these areas, the genes for these areas, but it's something you can read. I'm just showing you that a lot of these diseases that we're talking about have mutations at certain areas. Polycystic kidney disease results as a point mutation at chromosome number 16, PKD, polycystic kidney disease 1, or chromosome 4, PKD2, polycystic kidney disease 2. So as you can see, these genetic diseases arise because of mutations at some areas, and the areas are clearly known. And these areas are usually due to proteins that function, for example, hemophilia, which is factor 8. And point mutation means it's only one point in that whole protein where there's an abnormality. I'll pause again to inquire if you are following or there's something that I need to clarify. Kelsey?
One student has asked differences between PKD1 and PKD2.
Well, at this point, you are still you are asking me to teach you diseases. At this time, just know there is polycystic kidney disease. Those will teach you at a later point. Right now, the take-home message is to know that there's a mutation, and the mutation occurs. The kidney just becomes cystic. So the rest will teach you. For the time being, just know that there is a mutation.
Okay. Another student has asked the location of on chromosome.
What's that?
Okay, they didn't specify the location of what. Another student has asked the difference between penetration and variable expressivity in gene disorders.
So I'm coming to that. So these mutations, which we said were alterations to normal, can involve autosomes or usually the sex chromosome, but commonly the X chromosome. So a mutation is alteration from normal. And sometimes, as you saw that these alleles are two, there are those genes that appear as a recessive trait. Meaning that since we have a pair, we have similar gene on either side of the chromosomes. So those that appear as a recessive trait means that there has to be mutations on both. And remember that these genes, one is from the father and another one is from the mother. So for you to get a recessive trait characteristics, then there have to be mutations on both alleles. This is commonly seen in in proteins that act as enzymes. And there are those that come out as a dominant trait, meaning only one allele is having a mutation. This is usually for those that appear as structural proteins. In this, because one copy, the abnormal gene leads to formation of an abnormal protein that interferes with formation of tissues. Hence, in this dominant trait, even with one mutation, then you're going to have a structural defect. The pattern of transmission can be confounded by penetrance. Penetrance means there are certain mutations that specifically lead to disease. That association, for example, if you see a mutation in a gene and expression of disease, those, if it is one-on-one, then this is referred to as high penetrance. What this means is that there are certain diseases whereby you have a mutation in the gene, but in the expression, in the phenotype, you rarely see part of that. An example is, you will find, for example, somebody who has genes for brilliance, and yet doesn't show in the phenotype, in the expression, doesn't look bright. So in that, you can say these genes particularly have a low penetrance. Is that clear? Reduced penetrance means that the gene is present and can be transmitted to offspring, but does not produce the phenotype in the parent. This is this as opposed to variable expression, whereby the disease is present, but the severity of expression varies from mild to severe. So variable expression is a situation whereby, for example, somebody has factor VIII mutation and hence is a hemophiliac. But even in hemophiliacs, you'll find out that there are those that need lots of factor VIII and those that need less factor VIII. Such is what is referred to as variable expression. Does that make sense, or should I repeat?
Students asking if you could kindly repeat.
Exactly, that's what we talked about, penetrance. There are some of you who get it very quickly and some of you who get slowly. Despite all of you, we assume that medical students are bright. But as you can see, even in this lecture, there are those who get it pop and those that who take long. That is referred to as penetrance. Penetrance is a situation whereby a gene may not express itself despite being there. For example, I give you an example of genes for brightness, whereby somebody has genes for brightness, but when you look at them, they don't seem to be bright. So in such a situation, we will refer this as reduced penetrance. But in variable expression, the genes for brightness are there. When you look at the phenotype, there's brightness, but even as bright students, as a teacher, I know, a lot of you are bright, but there are those who get 80, and there are those of you who get 50. That is referred to as variable expression. Is it better? Kelsey, you are the only one I know because no one else is speaking. Unless you have a particular question, you can unmute. You can ask Kelsey to unmute you and ask. Okay, let me push on meanwhile, unless there is any burning question.
Okay, there's two questions here.
Yeah.
One is, can gene mutations in precursor cells be absent in differentiated cells?
Yes.
Okay, and the next one.
That's what I talked about. Repeat that question. Can gene? Repeat that question.
Oh, sorry. Can gene mutations in precursor cells be absent in differentiated cells?
No, the mutations are there. The issue is the expression, the phenotype, what the result is. You understand what it means? Remember, mutations are in the DNA, right? But the phenotype is what is the end result, the proteins that are formed. So let me just proceed and so that you can see in examples. Mutations can occur in many forms, okay? And remember that what we said, mutations is alteration from the normal. So a missense mutation is a change in a single amino acid. So in this, for example, when we went back, if I go back, let me go back so that you can see. If you see here, these three base pairs usually code for an amino acid, okay? If there's a change in one of these base pairs, then when it's being read in the ribosome, instead of CTG, if T is removed, then this is going to be CGC. So that change in a single amino acid is referred to as a missense mutation. So I go ahead again. Missense mutation is a change in a single amino acid. And remember, this single change will affect the resultant protein. And missense mutation have been seen in situations like sickle cell anemia. A nonsense mutation is a change in a stop codon. So amongst these three base pairs, there are certain forms that read stop. If you change one of these codons, then there is endless production of this protein. This change is referred to as a nonsense mutation. Deletion is a loss of a single base pair with a frame shift. Okay. So a missense mutation can involve either a single base pair or a big so-called whole amino acid. But if you have only a single loss, this is referred to as a deletion. It results to a frame shift because, as you saw, that long frame of reading is going to be changed because you have changed one single base pair. An insertion is again. A duplication is an extra gene with more protein production. So one frame, you have two of them. So you have more proteins, which is not necessarily beneficial. Splice site mutations are the ones which I explained to you, whereby an endonuclease cleaves not exactly at that boundary. So you find it has either gone more into the exon or more into intron. So those abnormalities are referred to as splice site mutation. Triple repeats. At the end of each chromosome, there is what is referred to as a triple repeat. And this is supposed to ensure it's like it makes the chromosome more stable. So if you have triple repeat mutations or increased tandem repeats at the end of the chromosome at each of the end, then you find what is referred to as triple repeat mutations. And these have an increased propensity to mutations, especially cancerous diseases. So these are examples of mutations. So if you are going to read and find that this is a deletion, for example, let me go back, like Duchenne's, it's a deletion at X chromosome P. I've left the locus because it is going to confuse you more. So in this Duchenne's muscle dystrophy, you find the main protein there is dystrophin. So dystrophin is one of the structural proteins. And when you have a deletion in part of this protein, then you get what's referred to as Duchenne's muscle dystrophy. So these people, you find the muscle starts becoming weak and it starts becoming necrosis. And these kind of patients, they have delayed milestones when you read more about Duchenne's. So a child reaches 5 years, and instead of walking and start running, they start crawling. They can't stand upright because of a problem in dystrophin. Where was the problem? There was a deletion of the dystrophin gene, which is on the X chromosome on the short arm. So that's referred to as a deletion. While there's a point mutation, for example, and this point mutation is most likely one of these, either a nonsense mutation or an insertion, any of these. So these are some of the things that you're going to read when you talk about single gene. Any questions?
Let me just scroll up a bit. So a question here, how will one differentiate between penetrance and dominant genes overriding recessive genes?
Pardon? Some of you are asking very hard questions, which I don't know if you understand them.
Okay, I'll just let that student rephrase. Let me, because there's many questions. Another question is, what factors lead to reduced penetrance?
It's sometimes due to the genes themselves. There are certain genes that are known to have reduced penetrance, and there are certain genes known to have increased penetrance. This is just about reading about how the gene manifests. The nitty-gritties I may not know, you may need.
Okay. Another question is, can allelic heterogeneity contribute to penetrance, or are they unrelated?
Allelic heterogeneity, some of you have read to a lot ahead, but I don't think it's so necessary. But allelic heterogeneity can affect penetrance, yeah.
Okay. Another student is asking,
why can't the students be simple? Because if you understand the simple things, then it will be easier for you. Because once they hard things, you'll understand them later. Some of the questions being asked are being studied at postgraduate level.
okay. Another student is asking if you could kindly repeat on triple repeat.
The triple repeats are mutations. You see at the end of the chromosomes, so here, the triple repeats usually found at the end of these chromosomes. And There are base pairs that are supposed just to signal the end. And remember, every time there's replication, there's shortening of that chromosome. So sometimes you find patients who have triple repeat mutations. So at that end, there's a certain amount of repeats that are required so that you know that the chromosome is ending. But in triple repeat mutation, you find extra. These are not necessarily beneficial. You find some of them are being associated with certain, especially malignancies. Remember, these are going to affect certain proteins, and some of these proteins are being implicated in, for example, malignancies. I think it's something that you need to repeat, to go and read about. And also do remember that whatever I'm giving you is not exhaustive. In the evening, you have to go and sit and read more. I'm just giving you the gist of what you need to know. But if you want to go deep, there are big books on genetics that you can even go and read more. Currently in this class, I want you to understand the simple concepts so that once you know the foundation, the rest of it comes in handy when you are reading about certain specific diseases.
Okay, thank you. Another student is asking what your frames are. No, sorry, what these frames are.
Frame. The frame is you see, for example, if I go back, you see like this is part of a protein, right? This is now a frame. This is how it's like you have stretched out the DNA, now you are reading the base pairs. That's the frame. So if you remove like G in this frame, like this G, okay? When this is now the DNA, if you remove the G, and now you are transcribing it to an mRNA, then you're going to miss out the G. So that frame has been shifted. You have, there's going to be total different reading. It will be read like CTC, T, GG. So it's totally different from the initial frame. That's what's called frame shift.
Thank you. Another student is asking, how can one differentiate between reduced penetrance and recessive genes?
Okay, now let's go back to, let's now go to patterns of inheritance so that you understand. Remember that these genes have to be transmitted from parent to offspring, okay? So as we saw earlier, in that base pair, in that, I mean, in the alleles, which are, you know, the chromosome are double. One is from the mother, and the one is from the father. Okay. Disorders that arise from inheritance is usually where you find whole chromosomes. Problems of inheritance are often seen in Mendelian disorders where whole chromosomes are affected. They can be classified as either autosomal recessive, autosomal dominant, X-linked recessive, and X-linked dominant. This autosomal, I've told you, it's from the autosomes. And the sex chromosomes, which are X and Y, you find a lot of disorders are usually on the X, more less in the Y. So, and we said that if it's a recessive trait, you need both alleles, both genes on both alleles being affected. Otherwise, if it's only one, then it's going to be referred to as a trait. So let's look at some of these conditions. Autosomal recessive condition, this pattern is typically seen with a mutation in a gene encoding for an enzyme, as I said, resulting in loss of functions. Homozygotes, who are people with both genes that are mutant, have such little protein, and hence there is going to be seen as disease. Heterozygotes usually have some that will make them still function as normal. And in this, as you are going to see in those patterns we used to read, the standard occurrence risk is 25%, meaning if the thing we are looking at is GG, if you put them as capital G and small g, when you do that combination, For it to be recessive, the small g's to be together, then the percentage risk is going to be 25% in that particular offspring. An example here is sickle cell anemia. Sickle cell anemia, or sometimes referred to as sickle cell disease, arises because of abnormalities in hemoglobin. You're going to learn about it. Hemoglobin is made of 4 globin chains that bind iron. Remember, iron is the main component of hemoglobin because of its oxygen binding capacity. So 2 of these chains are alpha and 2 of these globin chains are beta. And it's found that a point mutation in the beta globin chain in the gene, which is chromosome 11, leads to an abnormal globin. This abnormal globin causes red cells to sickle under low oxygen tension. And what is this point mutation? The point mutation causes a substitution of valine, which is an amino acid, for glutamic acid at position 6 of the beta globin chain. So if you go down to the globin chain and look at where it is, there is change of this amino acid, which is a glutamic acid. So you'll find that one of that base has been changed in such a way that valine is replaced. So the normal thing there is glutamic acid. So glutamic acid is removed and you put in valine. What does it lead to? That sickle, formation of the red cells. So you can see this is a point mutation, chromosome 11, coding for the globin, beta globin chains. So this is an example of a point mutation. What it means that the mother must be having either a trait or a sickle cell, and the father either a trait or a sickle cell, and when they merge, they find that the offspring has a sickle cell disease. Only a trait, somebody who has a trait usually will not manifest. They will just be normal. You will only know that this person could be having a trait either in extreme conditions of low oxygen tension or when they get an offspring with sickle cell disease. So in families, we find families, especially in Western Kenya, where sickle cell disease is rampant. A lot of people, before they wed or they want to partner, some of them do come for testing for sickle cell trait. So we look for this kind of so this is an example of an autosomal recessive abnormal genes for them to manifest as disease. This is as opposed to an autosomal dominant, for example, Marfan's. So Marfan's is an example of an autosomal dominant condition involving a structural gene referred to as fibrillin gene. This fibrillin gene encodes for fibrillin proteins. And these proteins are a component of microfibrils that form connective tissues. So in connective tissues, as you are going to read, I'm sure you do histology and anatomy, microfibrils are part of connective tissues. When you have abnormal fibrillin gene and hence abnormal microfibrils, then these people are referred to as Marfanoid. They have Marfanoid features because they are usually excessively tall, like some of you. They have long fingers, okay, something referred to as arachnodactyly. They have loose joints. So I'm sure you see clips of people in social media. Somebody can bend the joint abnormally, or they can do funny positions, acrobatic positions. Some of these are Marfan syndrome. So don't try it thinking that you're also that flexible. They also have ocular problems because the socket, the eyeball, is usually very bouncy or collapses very easily. And also they have this propensity for the vessels, especially the aorta, because of these microfibrils being very soft. So this condition is referred to as Marfan syndrome, in which the fibrillin gene, you need only one allele to have a mutation for it to manifest. Because even if both are abnormal, still the structural protein that is being formed is abnormal. And this is referred to as Marfan's syndrome. Any questions so far? Kelsey?
I'm checking the chats. It's mostly conversations between students.
Or tell them to avoid, because this is a class, tell them to avoid conversations within the class. We just want you to chat with us, with the teacher, so that you can understand. The others, you can do them outside class, kindly. So the other thing is X-linked recessive. And remember that X-linked is on the X chromosome. In X-linked recessive, in these patterns, females are typically carriers. The reason is, when you have two X's, one of the X is usually condensed as what is referred to as a bar body, so it's inactive. One of the X is inactive. So you'll find that most of these patients, because they need to be a recessive, then the females will not be affected because one of the X is already inactivated. So that leaves some of the functional genes available to produce product. They do not display the phenotype. But because of variable expression, you'll find that the people who are affected, you'll find that some of them have very severe disease or others have mild disease. An example of this is hemophilia A. Hemophilia means excessive bleeding, and it's also referred to as Christmas disease. It's an example of an X-linked recessive condition because of lack or mutation in clotting factor 8. So mutation in this clotting factor means the patient or the client or the patient is unable to clot normally. They have excessive bleeding. You remember that if you cut, let's say if you have cut, then the body puts in mechanisms to avoid that bleeding through the coagulation cascade, you know about it. But these patients who lack factor 8 means that now clotting is unable to continue and hence they bleed. But in these patients, if you go to the hemophilia clinic, you'll find that when we are injecting them factor 8, some require 10 units, others 100 units. This phenomenon is referred to as variable expression. The patients have the mutation, but in the phenotype, the severity of disease varies. So hemophilia A is an example of an X-linked recessive condition. X-linked because the gene is on the X chromosome, and hence, and it's a recessive gene because you need both to be, but for females, because X is already condensed as a bar body, then they have just enough proteins, they behave like traits. So males are the mostly affected. What about X-linked dominant? Theoretically, both males and females will be affected, but a double dose of the abnormal gene product has been found to be lethal in utero, so virtually no males are seen with the disease. So you rarely find diseases that are X-linked dominant because it's thought that once you have these, either these are the fetuses that are aborted because they are lethal in utero, or they die quite early and you never come to see them manifest. In all this, there are diseases that have a multifactorial inheritance, meaning no one specific gene is involved. It's either multiple genes and environmental factors that interact to produce a phenotype. Remember, in the initial thing that I showed you, let me just go back. I told you about this, that there are diseases that are 100% genetic, Down syndrome. And there are those that are 100% environmental. You're struck by lightning or you poison yourself with alcohol or whatever. Those are environmental, they are self-caused, there's no genes in that. But in between, for example, diabetes or cancer, they have a multifactorial causation or inheritance. These are the ones now I'm discussing with you, that these diseases have either multiple genes that are involved, or there's interplay with environmental factors. And the recurrence risk for this is approximately 3%. But it's difficult to state precisely. For example, diabetes. In the non-insulin dependent diabetes mellitus, you could be having a family history, hence genes. But if you know and modify your diet, avoid smoking, or avoid sedentary lifestyle, then you may not get this diabetes mellitus despite there being some genes involved. This is the so-called interplay between genes and environment, and this is what is referred to as multifactorial inheritance. Any questions so far?
Is there a difference between autosomal dominant and recessive?
Yes, that's what I've been explaining all along. Or do I repeat?
Okay, that one was sent a bit earlier. Maybe it was clarified. Another student has asked, what advice would you give to those who are ready to wed and have recessive sickle cell genes?
And they have recessive genes, or they have traits?
They have recessive genes for sickle cell.
Recessive means they are suffering from sickle cell, right? Or I don't understand what you mean. Recessive, okay. So if you have recessive genes in sickle cell, that means you have sickle cell disease. But if you have a trait, that means you're not manifesting. So if both of you, the partners who are coming together, have traits, then we advise you not to. We give you an advice. Of course, whatever you want to do after that is totally upon you, but we'll advise you. But remember, genetic diseases, they're a bit tricky because there are some people who are in love until they don't want to know about all these recessive. They'll want to take, they want to remember that even if you have traits, in a particular pregnancy, the chances of you getting a sickle cell disease child is what, Kelsey?
Pardon?
If you have a trait, sickle cell trait, okay, the mother and the father, in the offspring they get, what is the probability of the offspring having sickle cell disease?
25%.
Correct. So the chance of getting a normal child is?
75%.
Correct. So even after advising parents, there are those who want to take the 75% chance, but we give you the figures so that you can choose for yourself.
Okay. Another student is asking if you could kindly recap on X-linked recessive.
Wow, because you're giving me a lot of work. So some of them you need to go read. You are sharp guys. So you know about recessive, okay? Recessive means both chromosomes have to be having mutations for them to manifest. But in X-linked, remember, X is on the X chromosome. So females are usually XX. But remember, when you look at if you look at the karyotype of a female, you will not get both X's. One X is usually condensed as a bubble. It's a natural phenomenon. It's usually inactivated, which is a lucky thing, meaning that if that never occurred, then females will also suffer recessive conditions. Because of this X inactivation, there is some functional genes available to produce product. And hence, in X-linked recessive in females, they do not display the phenotype. That's what is meant by X-linked recessive. Females are typical carriers and males are typically sufferers.
Thank you. A student is asking, why is hemophilia A called Christmas disease?
Well, maybe that's part of their homework. They need to go and find out. but it is probably somebody, you might find it's somebody's name. I'm not sure, but you can read. Those are things you can read.
Another student is asking,
Yeah, ask.
If you could kindly recap on the basis of the lethal nature of X-linked dominant traits in utero.
Yeah, so X-linked dominant, when we talk about X-linked dominant, it means that when you have even one mutation on that X, then both males and females we are going to be affected. But it's been thought that because there are not many X-linked dominant, actually I didn't talk about Y-linked, there are also Y-linked conditions, and one of the common ones is referred to as hairy pinna, common in Asian communities. But it's also not such a big issue. But for this X-linked dominant, it's not commonly being seen, diseases with X-linked dominant. So it's been thought that the abnormal gene product, usually a protein, is associated with being lethal in utero, means the fetuses or the conceptus dies. So virtually, you find no one being affected. And read more about it.
Okay, the last question is, what did you mean when you said double dose in X-linked dominant conditions?
As in, if you find that both like, for example, in the double doses for females, you know, they have XX. It's been thought that if you have for both, even if one is there, then it should be able to manifest, but we never see it. And also the same as in males, because males have only one X. So these X-linked dominant conditions are literally not being seen.
Okay, thank you.
So with all those, what does it mean nowadays about these disease conditions? One of the things I've talked to you about is genetic predisposition. So genetic diseases, like somebody asks, if somebody has a trait or has a family history of sickle cell, when you get a partner and you want to get offspring, you can come to the lab and we test you for some of these genetic diseases. Also, there are those, let's say, mothers who have congenital anomalies which are genetic associated. So even in utero, we can be able to test if there is an abnormality with the fetus using what is called cell-free DNA. So what has been found out, you can go read more about it, is that when a fetus is conceived or there's a conceptus, part of the DNA of that fetus escapes through the placenta and you find it within the circulation. And nowadays we can get blood and then centrifuge it and get cell-free DNA and do a lot of genetic tests. So some of these are being done. The other thing is some diseases may have different optimal treatment. And this is being seen especially with certain cancers, whereby targeted therapies are now being given. For example, there is a disease whereby there's increased proliferation because of mutations that ensure that the protein product act like a growth hormone or something that ensures that the cells keep on proliferating. And now with targeted therapy, we have monoclonal antibodies that go bind on that site, let's say on the mRNA of that, of those, and hence stops the continuous multiplication. So diseases now have what, especially cancers, if you go read about targeted therapies, is because on the basis of these mutations which are being known. And also in future, it's being seen that there will be targeted treatment for doses of drugs, for duration of drugs, dependent on your genes, on how your genes react to this. So, and there's a lot of tests, a lot of studies that are being done on that, because there are some people who are slow metabolizers and others who are fast metabolizers, for example. Like if I know a lot of medical students drink alcohol. There's one who will drink one bottle and there's nothing. And there are those who drink one and they're already singing the loudest. This is because of alcohol dehydrogenase. So there are those who are referred to as fast metabolizers and slow metabolizers. So in these people, for example, if you are a slow metabolizer, then we can tell you, you take 1 and you are okay. And if you are a fast metabolizer, we can give you 3. So if you take that example towards treatment, you can start targeting the treatment dependent on the different behaviors of patients. The other thing is in clinical applications. We have seen that a lot of mutations lead to neoplastic conditions, and there are specific mutations that have been known to lead to certain neoplasia like leukemias. So instead of when you come to the lab and I show you how to diagnose, let's say, leukemia, nowadays you can even just do the genetic mutations. And once you get it, you know that this person has this problem. The way you see a Down syndrome's child, once you see those typical fasces, and you just do the karyotype and you know this, you don't have to take history. Come to the lab, we do the karyotype, we find trisomy 21, you know, this is it. So these neoplastic conditions and certain conditions can now be detected using knowledge of what you have learned today about the specific diseases. And part of it is here about the genetic diseases. The other thing is about identity testing. Most of us are not children of our parents, but we say so because they tell us. But when there are disputes, we are now doing DNA typing to be able to state clearly, are you an offspring of this so and so? The other thing that is being used is in crimes whereby certain genetic materials has been left in a site, and you want to know certain culprits who have been arrested, you want to know this belong to them. So you test for the DNA and match it with that particular individuals. The other thing is in identification of microbes, so that if you have a sample and you really want to know is it TB or not, there are specific ways using DNA, probes and so forth, and finding out, because TB, the genetic information of TB is now known. So if you get the nucleic acid probes and match it with what sample he has, and then you do what is referred to as PCR, you'll be able to know if it's TB or not. So this is the era of molecular diagnostics, and this is being done for many diseases. I think I'm going to stop there. So today, and these are some of the things that we are using. We are using things like PCR, genetic mapping, DNA typing, FISH, fluorescent in situ hybridization, CISH, chromogenic, immunohistochemistry, and many others. And even you as an individual, you can go to a genetic lab and have your genome typed and kept there. In case you get lost and there's some tissues which are suspected to be yours, they can be matched. And in future, remember that embryonic tissues are thought to be going to be used as spare parts in treatment of certain ailments. Like we talked about diabetes mellitus, where you can insert a gene into an embryo who didn't have an insulin producing gene, and they start producing insulin. So these are some of the things that are going to, that are coming up. But currently, these embryonic tissues being used as spare parts has not been approved because of ethical issues. Ethical in that somebody can take a very bad person and try to create themselves or clone, and this is not be ethical. I'm going to stop there. So today we have been discussing about the genetic basis of genes and genetic basis of disease. And we have gone through many things. Partly the main thing to know about are genetic basis of disease, which disorders have a clear-cut genetic basis. There are many, and I've listed for you some of them. And which diseases have purely environmental, that one you know, like accidents, whatnot, struck by lightning, those are purely environmental. In between, there are those that have a mixture of both environment and genetic. And these are some of the things that are now being researched on. Are there diseases which have more of a genetic basis than environment, and so forth. And we went through how these DNA to mRNA to proteins, how mutations occur, and how are you able to find out where the mutation is. So thank you very much. Now I'll pause for final questions.
A student is asking about sickle cell and malaria. Like, is it true if you have a sickle cell allele, or if you have sickle cell even and it's showing like you have the disease, is it true that it gives you immunity to malaria?
Yeah, that's true. And you will learn this more when you come to hematology.
Thank you. Another student is asking, how much does it cost to do complete genetic tests?
Depends on which to which. So you probably need to come to me if you want, and I tell you, but they're not cheap.
Okay.
Like screening, for example, if there's recurrent abortions in females, depending on what we are targeting, let's say, is it a trisomy 21, trisomy 18? So each of those maybe combined maybe 100,000.
Okay. Another student is asking, how long does it take for a mutation to become dominant in the population?
No, they need to read about what dominant is. They're talking about dominant and recessive. Those are patterns of inheritance.
Okay.
Do you understand? There's a difference between a mutation and patterns of inheritance. You see, if you have had a mutation, what is the probability for you to pass it on? So whatever we have been talking about, patterns of inheritance, how they move from one offspring to another. So don't mix the two.
Another student is asking the reference book for genetics.
There are many, but standard books usually, most of the standard books like pathology books, they will just give you a chapter on it. But if you have, if you really are interested in genetics, there are many, many books. But for the first one you can read about is about chromosome, and I showed it to you, that one, the human genome.
Okay. Students are asking if you'd be willing to share the slides with us.
Yes, I can share the slides, but you need to read more.
Okay. Another student is asking, if sisters have the same inactivated X chromosome, do they share the same traits?
Was that? I didn't get that.
If sisters have the same inactivated X chromosome, do they share the same traits?
Unless they're twins. Well, I didn't understand what they mean. But remember, every individual is unique. Like if you look at your sister, you definitely, you are from the same parents, but your genome is totally different.
Another student is asking about the cost for DNA testing for like paternal disputes.
For father and child, usually it's father and child. Okay, we can do 2-part or 3-part. 2-part, meaning usually it's fathers who don't know their children. So for that 2-part, like in my lab, that will cost 25,000. So we do the DNA for the father and then the DNA of the child, and then you match. So we do certain conserved chromosomes and then you match. So that's roughly 25,000. If it's 3-part, if you want to include plus the mother is in doubt also, so it raises the cost maybe around about 30,000-35,000.
Thank you. Another student is asking, what necessitates the use of non-invasive procedures today to detect any mutations? For example, amniocentesis, yet we have means of detecting these mutations non-invasively in maternal blood.
What necessitates that somebody uses invasive? It's knowledge. Basically, there are those who don't know that we have cell-free DNA, and there are those who know. So it depends on the knowledge and where you are. It's like asking, why don't Kenyans go to the moon? Maybe they don't have the means of going to the moon. But if you go to America, you'll go to the moon because they have the means. So if you're talking about the currently practice, you don't have to do amniocentesis because of the inherent risks of inducing abortion to this mother. So the main thing that is in use now is cell-free DNA. Take blood, get the cell-free DNA and do tests on it.
Okay. Another student is asking, can a person be born while he or she has reduced penetrance?
Pardon? I don't
Can a
think that's
person-
a question. I think to summarize this, there are a lot of terminologies there that you need to go sit and talk about, because I've had so many people talk about. One of the ones is patterns of inheritance, recessive, dominant, and so forth, mutations, penetrance, and variable expression. I understand the difficulties in understanding them from the word go ab initio, but there are a lot of books you can sit and read more. I'm sure some of you have not read into it, but once you read, you'll understand more. But from the basis of this lecture, you should be able to understand some of these terms, because I've tried to simplify the main things that you need to understand. In the interest of time, I think we need to stop there. I know it's supposed to be up to 12 or 1.
1.
But because you cannot, I don't know why it was put up to 1, because you'll not be able to concentrate if I continue. Use this other time to read more. There are a lot of, if you go to the internet, if you go to the library, there are lots of books on genetics, and you can start with the basics and then read more. And if you have any questions, you can mail me, or you can come to the department and I can be able to answer your questions in details. So go back, read more about these genetics and the genetic basis of this. What we want really you to take home is that the diseases, you as a doctor, an upcoming doctor, we are going to treat which have a clear-cut genetic basis. Understanding these diseases, having a genetic basis, one, helps you to explain to the patients why they are suffering from those diseases. Number two, there are certain diseases that have targeted therapies like cancers, and they are because of the genetic basis of these diseases. So you'll be able to manage them as well as possible. I wish to stop there. Thank you for your audience. Class rep, kindly take for me a roll call register for those who have attended, and you'll pass it to me in the department.